Thank you very much for the kind invitation. This is a great honor for me to be here with you and have the chance to present you Crispr-Cas9 at this very nice site where the Academy is located. This is a conference Louis Pasteur, so I was asked to choose a scientist to dedicate this conference. So I chose Louis Pasteur because he's French and I'm French, but also because he's a microbiologist. And as a matter of fact, I was educated at the Pasteur Institute. Having said this, I will show you some quotes of Louis Pasteur, which I find very interesting. So the purpose of my talk today is to explain you what is CRISPR-Cas9 as a technology, but more importantly, to explain you what is CRISPR-Cas9 in nature and how we came to work on CRISPR-Cas9 and how we came to understand the mechanism that has been harnessed as this powerful genetic technology. So first quote of Louis Pasteur, which I like very much because it's maybe 150 years old and I think it's still very modern. There does not exist a category of science to which one can give the name applied science. There are sciences and the applications of science bound together as a fruit of the tree which bears it. And since I'm an advocate of basic science, fundamental science, and I believe that any type of innovations anyway originates from basic science, I think it's a very nice quote. And as a matter of fact, this is what Louis Pasteur was doing. He was doing really basic science, and ultimately was really thinking always how he could apply the results that he was accumulating in applications. The CRISPR-Cas9 technology is represented here as molecular scissors. So you see the DNA here as a double helix that is targeted by this tool, here represented as scissors. And the CRISPR-Cas9 tool is a two-component system. It consists of a protein that is here represented as the blue scissors. So the protein is called Cas9. And this protein can cleave DNA in a programmable fashion. And what provides the programmability of the system is an RNA molecule to which the enzyme Cas9 associates. And this RNA molecule is actually the duplex of two molecules existing in nature. One molecule is called tracer RNA and the other molecule is called CRISPR RNA. And in principle, the programmability is designed as such. The RNA molecule can be designed to base pair specifically to a certain sequence of DNA of interest. And this is this complementarity between the guide RNA and the target DNA that allows Cas9 to target exactly the sequence of DNA of interest. And so this is a nice and simple way to have a programmable system that allows then Cas9 to really cleave the DNA at the site of complementarity. What is important for the system to work, besides this two-component system composed of a protein and an RNA molecule, it's a motif on the DNA that is called PAM, for protospacer adjacent motif. I'm not going to enter into what is a PAM because it's a bit complex, but in principle it's a short stretch of nucleotides. And in the case of this tool that is used worldwide and originates from a bacterium called Streptococcus pyogenes, this stretch of nucleotides, it's composed of NGG, and you can find a lot of NGGs on the genome, so it's not very restrictive. But however, for the system to work, this PAM has to be present downstream of the sequence that is targeted by the protein Cas9. So the protein Cas9 first scans the genome to find this PAM yellow sequence, and then with the help of this programmable RNA, will find the right DNA sequence to after be able to cleave the DNA. So, as it was said, it's a technology that has been adopted by the community of biologists very, very fast. And I will explain you later the different applications of this technology. We came to understand the last step of this CRISPR-Cas9 mechanism that allowed us to harness this mechanism into a technology around 2012. And for sure, in my laboratory, this research started actually already in 2007. But very fast after the description of this mechanism, the last step of the mechanism, and the description of how the biologists could use this mechanism as a gene technology for their cells and organisms of interest, very fast, so 6 months later, one could already see some publications showing indeed that the technology was working well as a precise gene surgery tool in any cell and organism. So this was quite impressive, and this is really to show how the technology is working well. And this led a year after we published our paper in Science to another publication in the journal Science entitled The CRISPR Craze, with a subtitle, A Bacterial Immune System Yields a Potentially Revolutionary Genome Editing Technique. So this paper has been published now 4.5 years ago, and indeed this was true. The technology is really largely used worldwide. And now I would like to bring this gene technology into an historical perspective. And for those who are biologists, to remind you, and for those who are not biologists, to maybe let you know that the rules of fundamental genetics were established in the 19th century. Here are some milestones, selected milestones that are shown, with Darwin in 1859, who for sure extrapolated on the origin of species. In 1865, Mendel, who was establishing the laws of segregation of alleles. In 1871, Friedrich Miescher, who isolated DNA. And one needed to wait something like 70, a little bit more than 70 years to have the understanding that DNA is a carrier of genetic information. Then in 1953, the structure of the double helix of DNA that was determined. And in 1966, the genetic code that was revealed. And surely the need to understand the genetic code and the need to understand the biological functions of genes and understand the complexity of genomes and their diversities. And this came in the late 60s and 70s. And a bit louder? Okay, I have to speak louder. I will try. So this came, this type of development of genetic tools that would allow really to understand the functions of genes and understand better the genomes came from research done on bacteria and viruses. So here I cite François Jacob, who was also a French microbiologist, and obtained the Nobel Prize together with André Lvov and also Jacques Monod in the 1960s, focusing their research on the understanding of enzymes and also on the understanding of viral structure. So exactly the same type of research that CRISPR-Cas relates to. Having said this, François Jacob had a very nice quote that is, nature is a tinkerer, not an inventor. And actually they were working on bacteria and viruses and understanding already that those bacteria and viruses will be a large source of enzymes that will be very useful as tools to act on the DNA and also on the expression of the DNA. And Jacques Monod in 1970 had also a very nice quote that was, modern molecular genetics offers us no means whatsoever for acting upon the ancestral heritage so as to improve it with new features, to create a genetic superman. On the contrary, it reveals the vanity of any such hope. The genome's macroscopic portions today and probably forever rule out manipulation of this sort. So although Jacques Monod was quite visionary, he underestimated let's put it that way, the power of the bacteria and viruses, having evolved a large number of mechanisms that have been very useful in molecular biology and genetics, and CRISPR-Cas is a very nice example of it. So in the mid-70s, a number of enzymes originating from bacteria and viruses were discovered and were harnessed for genetic technologies, so such as restriction enzymes, diverse enzymes that allow to recombine DNA, also enzymes that allow to transcribe the DNA into RNA, and so on. Also in 1977, the possibility to sequence the DNA, and later on in the 80s, the possibility to amplify DNA. And I don't highlight on this slide also all the technologies that have used the targeting of RNA molecules for knockdown approach. Now, having said this, the gene editing approaches came later on. So it started 20 years ago with the engineering of new types of nucleases, zinc finger nucleases, and also talon nucleases, that are engineered nucleases that provide exactly the same type of reaction that CRISPR-Cas, but they work a bit differently. But in principle, these are specific nucleases that can be targeted to certain sequences of interest on the genomes and cleave the DNA in a double-stranded fashion, the same way CRISPR-Cas9 is doing. However, for this type of nucleases, zinc finger nucleases and talon nucleases, one has always to engineer a new type of nuclease for a certain sequence of DNA to be targeted. And this is quite cumbersome for the biologist. So the programmability always relies on engineering a new protein, whereas CRISPR-Cas is easier. The programmability relies on the engineering of an RNA molecule, and that is easier to engineer than a protein. And the protein remains Cas9, and that would be the protein used to cleave the DNA. So this tool really provides a simplicity and also a faster way to modify the genomes and their expression compared to talon nucleases and zinc finger nucleases, and the specificity of the CRISPR-Cas9 technology, and that's why it has been adopted and developed by the scientific community so fast, is that it's a very versatile tool. So in itself, because of its way of working in nature in bacteria, it has been harnessed in various ways to modify the DNA and also the expression of the DNA. So the tool that is used worldwide is actually originating from the bacterium Streptococcus pyogenes, that is a human pathogen, strict human pathogen, so causing a large variety of diseases in humans. This is the main pathogen with which we are working in my lab. So just to show you, because one should not forget that this tool really originates from bacteria. So here you can see in red the cocci, the bacteria that are highlighted in red for the purpose of the picture. And they are invading human epithelial cells. So here these are human pharyngeal epithelial cells that are colored in green for the purpose of the picture. So these are really bacteria interacting with a human host and causing large variety of diseases. And in my laboratory, we are mainly interested in bacterial diseases, with really the reason why it's because infectious diseases remain the leading cause of death in children and adolescents worldwide, and the antibacterial resistance poses a major health threat. But when one works on bacteria, in our case, we are always interested in discovering new types of mechanisms that could lead to new ways to treat bacterial infections, so develop new anti-infective strategies. But when we work on bacteria, we know as well that we can always find interesting mechanisms that could be harnessed into genetic technologies, like it was shown in the past. Specifically, when one works on RNA molecules, that was our case. So, in principle, we are interested in different types of mechanisms that allow to understand how the bacteria as pathogens can attack the human host, and to a certain extent, how the human host defends itself against the pathogen. We work on different regulatory aspects of protein nature and also RNA nature, and trying to understand different types of regulatory mechanisms in the context of their role in the expression of virulence factors and other types of factors that the bacteria produce to be able to cope with their environment that is involving quite a number of stress in the human host, such as osmotic stress, heat shock, and that allow for us to understand how the bacteria can adapt to this hostile environment and can cause diseases or can persist in the human host. And this is with this global question that we came to CRISPR, and mainly with our interest to work on small RNA molecules. So DNA is transcribed into RNA that is translated into proteins, but it was discovered actually in bacteria in the beginning of the 80s that RNA molecules could have also regulatory functions, and this was already hypothesized, by the way, by Jacques Monod, could have regulatory functions and act as regulatory molecules that will interact directly with the messenger RNA or directly with the proteins to change the expression of this messenger RNA or protein. So the notion that RNA molecules are not only involved as a transition between the DNA and the protein, so this is a messenger RNA, or if you take ribosomal RNA or transfer RNA being really involved in protein synthesis, but this notion that RNA molecules can have regulatory functions. So in principle, they are in general small, and they can interact directly with the messenger RNA to activate or inhibit translation, or to also affect the stability of those transcripts messenger RNA. And they can interact directly with proteins to activate or sequester them from their function. What was missing when we started to work on small RNAs was to find small RNAs that will interact directly with the DNA and will change the fate of the DNA. And this is what CRISPR-Cas mechanisms bring. So this was a novelty we were interested in finding. We started a little bit more than 10 years ago in my lab. We were working on some RNAs having regulatory functions on gene expression in Streptococcus pyogenes, but we wanted to have a more global view of more regulatory RNAs existing in Streptococcus pyogenes that could have regulatory functions. So we used the bioinformatic approach to look for novel regulatory RNAs, and we found some novel ones that are highlighted here on the genome of Streptococcus pyogenes, and one which we called spRNA49 that is now known as tracer RNA. So we found this RNA, and we found that in the vicinity of this tracer RNA, there was also the CRISPR RNAs. And the CRISPR RNAs brought us to the CRISPR system, and at the time in 2006-2007, the knowledge of CRISPR systems was relatively limited. Having said this, what was known at the time is that here on this picture you have the part of the DNA that codes for this tracer RNA molecule, and what we found is that it was located upstream of a gene, and that was annotated to code for a protein that was a CRISPR system-related protein containing two nuclease domains, RUFC-like nuclease domain and HNH-like nuclease domain from Streptococcus thermophilus, another bacterium that is not causing diseases in humans but is involved in yogurt production. And this protein is now known as the Cas9 protein. So having said this led us to this CRISPR system, and we were wondering what this CRISPR system was. And in 2006-2007, the hypothesis was that this system was involved in the defense of bacteria against mobile genetic elements. So bacteria can act as pathogens and attack the human host or any other type of host, but the bacteria can also work as hosts themselves, and they can be attacked by mobile genetic elements of different natures. You can have the viruses of bacteria that are called also phages, and here in the bottom you can see the phages that can infect a bacterial cell with the DNA of the phage that is injected into the bacterial cell upon infection. And the invading DNA can replicate and can enter two cycles, either the lytic cycle where the DNA of the virus will replicate, will ultimately lead to the production of viral particles that will lyse the bacterial cell, so kill the bacterial cell and propagate to kill other bacterial cells. The invading DNA of the phage, when it enters the lysogenic cycle, the phage DNA can also integrate into the green genome of the bacteria and bring new genetic traits to the bacteria. And in this case, the genes that are brought by the phage become really part of the genetic traits of the bacteria. Another type of mobile genetic elements, these are plasmids, so circular forms of DNA that can carry genes that can code for virulence factors or that can code for factors that allow bacteria to resist the action of antibiotics. In any case, those genetic elements are mobile. They can be transferred from one bacterial cell to another bacterial cell, and from also sometimes different bacterial species. And the bacteria have evolved different defense systems to allow them to resist this attack of mobile genetic elements. Because sometimes it's good that they are infected by these mobile genetic elements when the mobile genetic elements can bring traits that are positive for the bacteria, but when it's detrimental, like, for example, the lytic phages, surely the bacteria have to defend themselves against cell death, so they have immune systems. You have different types of immune systems that bacteria have developed. If you refer to our immune systems that involve a classification of two types of immune systems, adaptive and innate immune systems. For the bacteria, this is the same. So all the defense systems that have been discovered up to now, beside CRISPR-Cas, are considered as innate immune systems. And one example is restriction modification systems that are enzymes encoded by the bacteria, so produced by the bacteria, and that can recognize the viral DNA and cleave the viral DNA. And these restriction enzymes have been harnessed as enzymes that allow to cleave DNA for the purpose of genetic technologies, and have been largely used for cloning purposes. So CRISPR-Cas is the first and only adaptive immune system which exists in bacteria, but also in archaea. It's adaptive because it implies a first recognition of the phage. So I will go through the different steps, but you have first a recognition of the phage upon the first infection that is necessary. And then there is a molecular machinery that is built and that allows to recognize this phage upon a second infection and destroy it. So I will come to the mechanism, but I want to give a brief history because this allows also to understand the different steps of the mechanisms that were discovered, historically speaking. In 1987, so more than 30 years ago, a Japanese group highlighted in the genome of Escherichia coli, that is a bacterium that is used as a model organism and as a host for cloning purposes in laboratories. One Japanese group discovered a series of repeats in the genome of Escherichia coli, so short sequences of DNA that are repeated. So they are here represented in black. And that were interspaced by sequences, unique sequences, short, of different nature. Now, at the end of the 90s, And beginning 2000s, what was described is that this series of repeats could be transcribed into RNA molecules of different sizes. So there was this notion that there will be an RNA molecule involved, but also an RNA molecule that will undergo processing, cleavage, to lead to short RNAs that were called CRISPR RNAs. So surely the history is more complex and it has involved many more scientists, but I try to simplify. And then what was shown at the beginning of 2000s is that in the vicinity of these repeats, series of repeats, there was always a series of genes encoding proteins that could have homology with proteins that were already described to act on the DNA or RNA, meaning proteins that can bind DNA, that can bind RNA, that can cleave DNA, or that can cleave RNA. And then what was shown later on is that these unique sequences that are located in between those repeats were actually matching sequences of mobile genetic elements, such as plasmids and phages. And then the hypothesis came that most likely this system will be an adaptive immune system existing in bacteria, but also in archaea, whereby there will be a step of memorization of different types of infections with different types of mobile genetic elements, and that those infections will be memorized by incorporation of sequences of mobile genetic elements into the CRISPR array at the level of this locus, and that there will be, after a molecular machinery in place, that would involve transcription of this CRISPR array so that the memorized infection will be ultimately contained into this RNA molecule that will associate to the Cas proteins and to then target the genome of the virus upon a second infection. And so the idea came also that CRISPR-Cas will be then an adaptive immune system existing in prokaryotes, and this was by analogy to also the fact that RNA molecules will be involved, and that this will be also analogous to RNA interference existing in eukaryotes, whereby small RNA molecules can target messenger RNA of genomes. So the idea is as follows, and there was a breakthrough paper in Science published in 2007 by my colleagues Philippe Horvaths, Sylvain Moineau, and Rodolphe Barangou, whereby a phage infecting the bacterial cell injects its DNA. There will be some proteins, so CRISPR-associated proteins encoded by these genes, and that will recognize the invading DNA, cleave a portion of the invading DNA of the phage to insert it into the CRISPR locus at the level of the promoter. And this will work as a memorized marks of former infections by the phage. And then this array is transcribed into long RNA molecule that is cleaved into short CRISPR RNA molecules. So each CRISPR RNA molecule contains the memorized portion of one specific phage. And if, in this case, the phage infects the bacterial cell a second time, then the complex of CRISPR-associated proteins that are associated to this CRISPR RNA can recognize the phage DNA and cleave the phage DNA. And if you cleave the phage DNA, then you cannot have replication of the phage, and this is the dead end for the virus that cannot propagate any longer. So this is the way the bacteria defend themselves against their infections. When we started to work on the system, what was already clear by the work of bioinformaticians is that the CRISPR-Cas system has evolved into multiple systems. And this is mainly based on the diversity of sequences. So you always have the CRISPR-associated genes, but the number of CRISPR-associated genes, also the nature of CRISPR-associated genes in terms of the proteins that they translate, so this nature is different, as well as the nature of the sequences of the repeats is also different. So you always have the CRISPR-associated genes, the CRISPR array, but the sequences are different. And this allowed a classification in different classes and different types and subtypes. When we started to work on the CRISPR-Cas9 system in 2006-2007, my colleagues were focusing more on the mechanisms of the type 1 and type 3 systems. And those systems were clearly involving a complex of CRISPR-associated protein guided by this CRISPR RNA. We had the chance in Streptococcus pyogenes to have the type 2 system. And the type 2 system is simpler. Instead of this series of CRISPR-associated proteins that are involved in the recognition of the phage upon a second infection to destroy it, those complexes of CRISPR-associated proteins are replaced by one protein that is called Cas9. So coming back to Streptococcus pyogenes and the two RNAs that we had identified that allowed us to come to the CRISPR system, what we showed is a totally different mechanism. So the dogma at the time was that one needs the CRISPR array encoding these CRISPR RNA molecules, and one needs these CRISPR-associated genes encoding the CRISPR-associated proteins, and the system is self-sufficient. One does not need any other type of molecule. From our screens on small regulatory RNAs, we had identified tracer RNA. Actually, we worked for 2-3 years in my lab trying to find the function of tracer RNA. We saw that it had another function until the point where we realized that it did not have another function. And so the idea that I had is to ask the question whether this RNA molecule will not have a role in the regulation or in the activity of this CRISPR-Cas9 system. And indeed, we showed that this molecule is essential for the system to work, and also involves a mechanism that is different from what was a dogma, that was that CRISPR-Cas systems are a complex of CRISPR-associated proteins guided by CRISPR RNAs. So what we showed is that this RNA molecule tracer RNA has the ability to base pair with the repeats of a long precursor CRISPR RNA molecule to form a duplex. This associates to the protein Cas9, and a molecule from the bacteria can recognize this duplex, cleave these duplexes to lead ultimately to the production of each CRISPR RNA that each contains the mark of a specific mobile genetic element. And the idea that we had is that this would not be the only role of tracer RNA to trigger the maturation of this long CRISPR RNA molecule to lead to this mature CRISPR RNAs, but that tracer RNA will remain bound to the CRISPR RNA, and that the system Cas9 will be very specific. It will be actually one protein guided by a duplex of RNA. And so indeed, this is what happens. And here you have the target DNA with this PAM sequence that has to be present on the genome. So Cas9 first scans the genome to find this PAM sequence. And then when the system finds the sequence that is complementary to the CRISPR RNA, here you see the complementarity, then the enzyme undergoes different stages of differential conformation. So this was also an hypothesis that we had, and that ultimately can cleave the two strands of the DNA at the site of complementarity, and using two nuclease domains, an HNH motif and a RUFC motif. And what happens is that a double strand break will be introduced into the DNA based on this RNA targeting. So this is what was described in the science paper. And so this last step of mechanism, also with the idea that we could even simplify further the system by linking those two RNA molecules via a linker, and the possibility for the biologist to design a single guide RNA. And the only programmability that is involved here is to replace the green sequence by the sequence of interest, depending on where the biologist would like Cas9 to act on the DNA. So the technology revealed to be extremely efficient. And what the technology does, because it's important to understand, the technology just triggers the break. The rest is done by the cell. Specifically in eukaryotic organisms, you have repair pathways that are quite efficient, depending on the types of cells. And when one introduces a break on the DNA, those breaks can be repaired via two different pathways. Either the so-called non-homologous end-joining repair pathway that is to a certain extent error-prone. What will happen is that there is a break on the DNA, and the repair system will introduce sometimes some errors that would allow to delete a portion of the sequence at the site of the break. So this allows to delete a small portion of the DNA or larger portion of the DNA, depending on how one designed the technology that targets the DNA to introduce the break. And if one introduces into the cell a so-called homology donor, meaning a piece of DNA that has homology to the DNA sequences that are upstream and downstream of the break, one can replace a certain sequence of interest by another sequence of interest. So via this means, one can introduce a specific mutation on the DNA, one can correct a mutation on the DNA, one can replace a DNA sequence by another DNA sequence. So we can do a lot of different tricks of the DNA. The idea that I had initially and the chance that I thought we had in the lab to work on Streptococcus pyogenes with this minimal Crispr-Cas9 system that works with one protein and a duplex of RNA that can be designed as one RNA molecule is that it will be very attractive for the community as a genetic technologies. And indeed, this is what we showed ultimately. And now, actually, it's clear that the technology has many potential applications in the life sciences, in biotechnology, agriculture, and medicine. So very fast, it was shown that it was working very well in plants, in human cells, in different types of model organisms of animal nature, in fungi, and in bacteria. The advantages of the technology compared to the technologies that were used prior to CRISPR-Cas9, such as zinc finger nucleases and talon nucleases, is that it's cheap, it's easy to program and to design. The system originating from Streptococcus pyogenes is very efficient. And what is important to mention is that the reason also why it was applied very fast is that the natural system is working already very nicely. So surely for the biologists, they know in bacteria there is no nucleus, so we needed to fuse the Cas9 protein to a signal that allows, when it is used in eukaryotic cells, to target Cas9 into the nucleus, and to change the codon usage so that the protein can be well translated into eukaryotic systems. But these were the only changes that were made, and the natural Cas9 protein was working very well. The technology is versatile, and I will show you some examples. And what is really also attractive with the technology is that one can do some multiplexing. The idea that one can design different RNA molecules and target at once different portions of the genomes. The system has low toxicity, which was also the reason why it was attractive. And surely there have been a lot of improvements of the technology with regard to the specificity, so that the technology and the tool can specifically act on a sequence of DNA of interest and not by mistake act on another piece of the genome. There also have been a lot of improvements with regard to delivery, because although the technology may work maybe even with 100% specificity, for certain applications, there are still some issues with regard to delivery systems that may not be so safe or so efficient. So there are lots of developments with regard to delivery technologies that allow to bring the CRISPR-Cas9 technologies to the right type of cell or tissue. And also because it is easier to favor with a technology some small deletions of the genome than to replace some sequences by other types of sequences. So there are lots also of developments to try to improve the technology with regard to gene replacement or introduction of specific mutations and correcting mutations. Having said this, what is interesting, and I'm not going to enter into the details of these slides, actually, but these slides allows me to tell you that not only you can really modify the DNA, but the beauty of the natural system, such as the Crispr-Cas9 mechanism and the way it works, is that you can also develop the technology to modify the expression of the DNA to target not only DNA but also RNA, and allowing to study, to modify the DNA, to study epigenetic marks, to also label the DNA and being able to trace the DNA. So this is what is unique with this technology, because it's a technology that everyone is using, but at the end of the day, biologists were also using restriction enzymes for a long time. So it's not the only tool which is really part of, how do you say, and will become more and more, let's say, an obligatory tool in the toolbox of biologists. But what is really specific with Crispr-Cas9 is that it's very versatile. And following the version 1.0, you add 1.1, 1.2, and so it's really a natural mechanism that allows to really perform multiple tricks on the DNA, RNA, and their expression. A very nice point with regard to Crispr-Cas9, and I will come to slowly to the applications and then end my talk. And what I like very much is the fact that it's a system that exists, at least the Crispr-Cas9 system exists mainly in bacteria. It has been found only in one archaeal species. Other types of CRISPR-Cas systems, so those that are working in a more complex fashion, or new ones that are also quite simple and that have been discovered lately, are present in bacteria and archaea. When we started to work on CRISPR-Cas, what I liked very much with this type of research, so it was already in 2008, 2009, is that we understood that the diversity of the CRISPR-Cas9 system would allow us to understand what is conserved and allow us to understand really the basis of the mechanism. So if you look at different bacterial species, and we found the system in more than 350 bacterial species, so largely found in nature, and in bacteria that can be found in different types of niches and environments. There is always some diversity into the architecture of the CRISPR-associated genes for the CRISPR-Cas9 system, and this applies also for other types of CRISPR-Cas systems. But if you compare the sequences of the Cas9 proteins, you also have a large diversity, and you have a very low percentage of amino acid identity, and the conserved domains are those that are important for the protein to work. And you have also a large diversity of RNA molecules in terms of their sequences, so this duplex of RNA molecules, but also in terms of their structure. And this is important because as a genetic technology, one would like to find Cas9 systems that are more efficient. I mean, the one of Streptococcus pyogenes, actually, this is the one that so far has been shown to work the best. And this was our chance to work on this bacterium that offered us the Cas9 system that is the most efficient in terms of genetic tool to be applied for diverse types of cells and organisms. But there is also an interest when one works on genetic tools to have a genetic tool that is very short, very small, to facilitate the delivery to the nuclei of different types of cells and organisms. So the nature has also evolved with Cas9 proteins that are shorter and longer. And there are a lot of developments to find variants of Cas9 or Streptococcus pyogenes that are more efficient and more precise. And also some variants that will be smaller. So a lot of efforts are done in this direction. But what I like very much is that it's a system that originates from evolution, and at the end of the day allows to study better the evolution and the diversity of biology, in biology, in the sense that it was cumbersome to really, and before zinc finger nucleases and talon nucleases, it was not possible to modify the genes, for example, of human cells. So now with CRISPR-Cas9, it really brings a higher level of possibilities for this type of modification. So it's very important to understand the functions of genes, and specifically in the context of health and diseases, but also in the context of life sciences in general. And this, it's interesting because a lot of studies in biology have been done either using mouse models or always studying the same type of human cells that are human cell lines used in the laboratory. So here we have the possibility, even though it's not always easy, but to work with primary human cells that are important for all the context of medical research. For sure, it helps in facilitating the genetics that is important to understand the biological pathways, to engineer better models for human diseases, specifically the engineering of humanized animal models. Further in medicine and health, so surely it's really used and developed for drug development. So it facilitates all the genetics that is used for finding new targets for therapeutics, and all those engineering of models that allow to validate the therapeutics in development before going to clinical trials. As a direct medicine, there are developments of the technology to cure human genetic diseases. The technology is also very attractive in the field of cancer immunotherapy, whereby immune cells are engineered to be able to recognize cancer cells and kill them. So this is a new strategy, and this involves genetic engineering, and CRISPR-Cas9 is helping a lot in this strategy. It's also a technology that is for sure very important in agriculture and the production of biological fuels. It facilitates the engineering of crops, also the understanding of mechanisms existing in plants. It really provides a technology that is more precise for crop engineering. So it's very attractive in this direction. And also, and this was the first application, this technology is the possibility to produce probiotics that are resistant to phages, and also very helpful to engineer industrial macroorganisms. Again, for sure, in the application of life sciences for research purposes and basic science and research and development, the technology is very attractive. There are surely some ethical issues with regard to the development of the technology in human embryogenesis, or to use in human embryos. So there are a lot of academies who are working on this aspect to work on different types of regulations that one would like to see in place to avoid the wrong applications of the technology, for example, in human germlines or in human embryos, if it was not for research purposes. In agriculture, as I said, it really allows to edit genes in major crops. It allows a faster and easier introduction of desirable traits. And it provides also less genetic baggage with CRISPR-Cas9 and other programmable nucleases. So also what is interesting is the definition of GMOs, which this definition implies the presence of foreign DNA in the plant that has been genetically modified. With CRISPR-Cas9, one can modify the genome of a plant with, at the end of the process, no foreign DNA, which for sure challenges whether this plant can be called a genetically modified organism. I already talked about the diseases, but really the challenge is really to use CRISPR-Cas9 as a direct therapeutic. So you have some first applications of clinical trials that have started, so combining gene therapy and cell therapy to treat certain blood disorders, for example, so where the idea is to really collect the cells of the patient, modify, correct a gene with the CRISPR-Cas9 technology, and then, for sure, verify that everything is fine, and re-infuse back the cells into the patient. You have other types of CRISPR-Cas9, of CRISPR-Cas system that have been discovered after we worked on the CRISPR-Cas9 system. We have worked on this CPF1 system that involves an enzyme that is also very interesting. Others have worked on this enzyme C2C1, C2C3, C2C2. You have a large diversity and always the possibility to harness these CRISPR-associated systems as technology, even though everyone agrees to say that the CRISPR-Cas9 system from Streptococcus pyogenes is very efficient. And for sure, a lot of biology is still going on from my lab and other colleagues, trying to understand the diversity of these systems and the new mechanisms involved by the new CRISPR-Cas systems with regard to the memorization step and the expression step and the targeting of the phage, and also an interest in trying to find new types of defense systems in bacteria and archaea that could lead to novel gene technologies. I would finish by thanking the different institutions where this work was done. I started with my lab members to identify this tracer RNA molecule and CRISPR RNAs when I was in Austria, the University of Vienna, Max F. Perez Laboratories, and this is really on my move to Sweden, the Laboratory for Molecular Infection Medicine Sweden, that is at Umeå University and is also a partnership with EMBL, the European Molecular Biology Laboratory that is mainly based in Heidelberg. So this is really in this institution that I came up with really the idea to link this tracer RNA to the CRISPR system, and that with my lab members we started to understand more the mechanisms. Then I moved 5 years ago, and I have to thank the Helmholtz Association. I was first in Braunschweig at the Helmholtz Center for Infection Research with an affiliation at the Medical School of Hannover, and 2 years ago I moved to Max Planck in Berlin, and I have an affiliation with the Humboldt University. And surely different types of funding, and specifically the Alexander von Humboldt Foundation that has been very helpful, and the Max Planck Gesellschaft with regard to funding my research. Surely my lab members, very talented junior researchers. It was a pleasure for me to really start this new topic in my lab as someone who didn't know anything about CRISPR, and certainly also as very young scientists, so a master's student and a PhD student. And this was a pleasure to really go step by step and discover together how the CRISPR-Cas9 system was functioning. A collaboration with a group of Jörg Vogel in Würzburg in Germany for the first step of the CRISPR-Cas9 mechanism. Then a collaboration with a group of Jennifer Darna in Berkeley for the second step of the CRISPR-Cas9 mechanism with Martin Jinek, who has now his own lab at the University of Zurich. And the group of Eugenie Kounin at Bethesda in the US, who has helped us a lot with the bioinformatic analysis and understanding the evolution of the Cas9 enzymes. This is my lab a couple of months ago in Berlin. I need to cite for conflict of interest 2 companies that I have co-founded with 2 partners, Rodger Novak and Sean Foy, so ERS Genomics and CRISPR Therapeutics. CRISPR Therapeutics is focusing on developing the technology to treat certain human genetic disorders. And I will finish by some other quotes for Louis Pasteur, because it's a conference dedicated to Louis Pasteur. And I think I like also very much these quotes that are very modern, I believe. In the field of observation, chance favors the prepared mind. Let me tell you the secret that has led me to my goal. My strength lies solely in my tenacity. Persistence in scientific research leads to what I call instinct for truth. One should doubt in experiments until the facts do not force to give up any doubts. And I would like to thank you for your attention.
Thank you very much, Emmanuelle. You showed how powerful genetics and molecular biology can be to tackle very complex systems.
Yeah.
Complexity is exploited, so to speak, in a way that you showed so well. I think you probably be available to take some questions. And I would like to ask the people that want to ask questions, please ask short questions. Don't start telling what you have done before and afterwards. Just ask the question so that Emmanuelle can answer short answers, and so that more people can enter in the game. Please, there are some questions. Yes. Give him the microphone. Morgante. Who else?
Have phages set up systems to avoid the CRISPR-Cas system?
Yes. Surely. In the arms race between viruses of bacteriophages and bacteria, surely the viruses have evolved systems to escape. So viruses have evolved systems that escape the defense systems of the bacteria. And the reason why is also that phages are outnumbering bacteria by a factor of 10. So these are actually the most abundant organisms on the planet, specifically in the ocean. So actually, phages are smart, so they can produce proteins that will interact with the Cas proteins, inhibiting them from their functions. They can also evade the CRISPR system by mutating the sequences of the DNA that is recognized by the RNA of the CRISPR-Cas system. So it's also a way to evade. And this PAM sequence that is essential for first recognition by the CRISPR-associated protein can also mutate. So via mutations or via production of proteins that can inhibit the CRISPR-Cas system, they can evade. So these are examples.
More questions, please? Sì.
Good afternoon. Sunday is the International Day of Women and Girls in Science. Is it difficult to be a scientist for a woman?
No, I think it's difficult to be a scientist, whether you're a woman or a man. I never asked myself the questions, actually. I was just a scientist. Then after I go my way, like any other scientist. I have had also colleagues, male scientists, for whom it has not been that easy. I have to say that what was more difficult for me was more to be a foreigner, not specifically in the US, but in Europe. Now being a foreigner and a woman can be an advantage, because a lot of institutions like to show that they have international women in their institutions. So at the beginning you're forgotten, and then at some point people realize that, oh, maybe what you're doing is quite interesting. And then at some point you go your way, and you show that your lab is productive and can do some meaningful research. And then after, the institutions are happy, and then they show you to the world.
Thank you.
But I think it's really difficult whether one is a man or a female. Now, I try to not really think about it, because otherwise there are some aspects that can be more difficult, specifically when women in science have a family. It's difficult to... But nowadays in Europe, you have also some programs, not some programs. Actually, I always hated programs for women in science. So when I was in Austria, there were some programs for women in science. I refused to apply to those programs. But having said this, now there is always an understanding that it is important if, for example, for a job application, a woman and a man is equally, ranked, that there will be an effort to allow the woman to have the position. But then after it creates also some issues, specifically in Germany, I saw it with this positive discrimination, with some colleagues having difficulties to, also some male colleagues having difficulties to get positions, because now the effort is on trying to promote
one more
females in
question there.
positions.
Giancarlo?
Are there homologous of the system in eukaryotes?
Yes, so in eukaryotes you have different, what is called RNA interference systems existing. So meaning RNA-guided system that can target messenger RNA. So this is what is called RNA interference. And you have different types of RNAs. You have the small interfering RNAs. You have the macro RNAs. You have also the pWe RNAs. And the CRISPR system will be more close to the pWe RNAs that are also involved in recognizing transposons, so mobile genetic elements of eukaryotic cells. But as a system that works, such as a CRISPR system that is adaptive, that involves these Cas proteins guided by RNA molecules, does not exist so far in eukaryotes. No one could find homologous Cas systems in eukaryotes.
Fabiola. Bolognesi.
The system is very elegant but complex at the same time. So in principle, it should be prone to errors. Can you comment on the error rate and where it lies?
So for sure, the system is a system that is existing in bacteria, and in bacteria, the system does not need to be 100% specific, because at the end of the day, it's just within the context of arms race between virus and a bacterial cell. If a bacterial cell is killed, other bacteria will survive. So this is one thing. Now, what has been shown is the Cas9 can be, has been developed now into a technology that can be 100% specific. But this depends a lot on how one designs the Cas9 technology. So first of all, if one is really bound to a specific sequence of DNA to modify, it may not be a sequence of DNA that will lead to 100% specificity. But otherwise, it depends also a lot of how the RNA is designed, the production also, the amount of RNA versus Cas9 protein. The system of asparagines is very efficient. If you add too much protein, it will not help, and the protein will try to recognize sequences that it's not supposed to recognize. So it depends a lot of the ratio between RNA and protein, the amount of RNA and protein, also the delivery system that is used, and also it depends on the type of cells. Every type of cells has a different, how do you say, the repair machineries work differently at different levels. So it's always specific. There is a general recipe. One has always to play around depending on the sequence that is targeted and depending on the type of cells.
Professor Parisi?
In the case of mammal immune cell, the T cell learn during chemic education to distinguish the self by non-self. How it works in the CRISPR system that the CRISPR does not attack the own cell?
Yes, this is indeed a point, and maybe I have to have this. Is this drawing okay? I'm not completely sure, but maybe it is. There is this PAM sequence, and this is the PAM sequence that allows to differentiate between self and non-self. Having said this, once the piece of phage DNA has been inserted into the CRISPR array, it's non-self at that point, but it's become self here. So one has to imagine here you have the green sequence that will end up being incorporated into the CRISPR array and transcribed into this RNA molecule. And the PAM sequence is in the vicinity. So the PAM sequence is important for the system to work, but it's important at the level of recognition and upon a first infection, and also upon a second infection. But what happens is that the PAM sequence is present on the phage DNA, but it's not present on the DNA of the CRISPR array. It's replaced by the repeats. So here, the mature CRISPR RNA with the Cas9 protein could not target the DNA at this level because the PAM sequence does not exist. So it allows the system to not cleave itself in a way and cleave only the so-called foreign DNA.
Prego.
Do you have any model for the evolution of Cas9 in the sense of before cutting DNA perhaps was just repressing gene expression, and before interacting with DNA was interacting actually with RNA? You know, if I posed rightly the question. Because you can modify Cas9 with just a single nucleotide amino acid, and then you get this defective one. So this suggests to me that it's possible that during evolution you had Cas9 working just on DNA gene repression, and then evolved the ability to cut.
I would not believe so. The trick to really harness Cas9 as a system that would target the RNA, it's something that will be difficult to, I think, to see in nature. However, so the HNH domain is really a domain that cleaves DNA, and the RUFC domain in general, it works like an RNase H, so it cleaves the RNA of a duplex DNA RNA. So in this aspect, it is possible that Cas9 was cleaving RNA at some point, but not specifically in the context of the way it has been designed to target the RNA, which here it's not modifying the enzyme, but more engineering, let's say, a DNA guide so that it can act on the target RNA. So the other way around of the natural way, that is to target DNA with an RNA guide.
One more question, please.
Could you tell us a little more about the strategies which are being pursued concerning delivery in humans?
The strategies, so for direct delivery, so there are some developments with regard to combining gene therapy with cell therapy. So this is the ex vivo approach. And here surely there are a lot of efforts to develop delivery technologies adapted to different types of cells to be targeted, to be able to correct a mutation. Because also one has to consider that these delivery technologies have been developed over the last 20 years, even maybe more, and that the field of gene therapy has evolved into gene editing field. Gene therapy before was always involving the introduction of foreign DNA prior to gene editing, and that the delivery technologies needed also to be now adapted to these gene editing techniques such as zinc finger nucleases, telenucleases, and now CRISPR-Cas9. So all the delivery technologies rely mainly on electroporation, so of human cells, on all this type of viral deliveries, so using this AAV type of vectors or lentiviral types of vectors, and nanoparticles. And with regard to an in vivo approach, that is for sure much more challenging, the idea that one could deliver directly the technology into the patient. So it's more actually the nanoparticles that are developed in this sense, but it's extremely challenging because the technology CRISPR-Cas9 has to be 100% specific, and the delivery has to be also extremely safe and efficient target the right number of cells.
One more question, then we take another one only because it's getting late.
So thank you.
Please.
So maybe you have
Speak
already.
up.
Maybe you have already answered. So how efficient is the system? I mean, most of the time you will not have a selectable marker to follow the homologous recombination or non-homologous enzyme. So how efficiently you can follow the correct modification?
In eukaryotic cells?
In eukaryotic cells.
But it's mainly by sequencing in principle.
Yes, but how many clones I have to sequence?
So it depends on the type of cells that is targeted, but indeed it can be as low as 1 to 5%, and it can be higher depending on the types of cells. I mean, actually, you can use also other methods than sequencing, but I mean, ultimately, to select for this. How do you say the fact that you will have a deletion, but ultimately at the end of the process, if you have a clone where you have that has been selected to be modified by CRISPR-Cas9, you will have to sequence to make sure that the modification has been done very much specifically.
One more question here.
Trying to correct a mutation in the human genome, do you think there are some factors, subcellular factors, that can influence the specificity and the efficiency of a CRISPR-Cas9 system? For example, DNA methylation, some particular cell cycle phase?
DNA methylation, not.
Basta, last.
Not to my knowledge. At least this was a question that we asked in my lab very early on. So we did some experiments to figure out whether DNA methylation was important, in the sense that in bacteria, if the foreign DNA is not always modified to fit the level of modification of the natural DNA. So, for example, if you take I can say an example, Staphylococcus aureus can accept DNA from Staphylococci, but not from E. coli. So you need to first do your manipulation in mutant bacterial strain that is restriction minus modification plus, that can modify the DNA to then bring the DNA that has been modified into another bacterial strain. So I don't know if I'm that, how do you say, clear? But in principle, we tested for the system in S. pyogenes whether modification of the DNA was influencing the efficacy of the system, but not. And I think, to my knowledge, no, in principle, in eukaryotic systems, the DNA does not have to be modified in a specific way. Cas9 will cleave the DNA of eukaryotic cells.
We have the last question, Professor Schiaffino.
Perhaps a crazy question. Can one envisage that this system can be used in some form of bacteriological war? For example, inserting this system into a virus, pathogens for humans, attacking a gene essential?
No, not that I think of. I've been working with bacteria for a long time, and bioterrorism linked to the fact that a specific virus or a very hypervirulent bacterial strain could be spread in the world. This is not new, and the possibility to engineer genetically bacterial viruses, one can really do so for a number of years, not using CRISPR-Cas9. So if really one had to fear these applications in bioterrorism, I think we would have seen it already. So CRISPR-Cas9 is not going really to increase the risks that someone would engineer a very nasty virus or a very nasty bacteria. And CRISPR-Cas9 even delivered via a virus that will infect human hosts, then you would have to have a CRISPR-Cas9 system that targets a specific gene, but which gene and you know, an essential gene, yes, but I don't think that it will be that efficient in this case. I don't see it possible. It's a scenario that is that I don't see that that is still science fiction for me. But
he's always asking anything.
Okay, okay.
So before closing, I have the pleasure of giving you. It's not gold. Medal for you to call this.
Very good.
This
That's very nice.
day.
Thank you very much.
I have something very special for you. There is a very limited edition of this small book, which is as a writing by Galileo. Writing by Galileo, and side by side the translation, which was made by a friend of ours, a colleague, Giovanni Bignami, an astrophysicist that unfortunately died last August, and it's a very special limited edition. I think since you have a lot of free time, you can
yes.
try to read it, okay? Thank you.
No, this is just when in my office,
exactly.
when people think that I'm working and actually I'm reading.
Galileo.
Okay.
Thank you very much.
Very nice, thank you.